Hi, everyone. Today we're talking about monohybrid genetics problems. But before we get into that, we should probably talk a little bit about the history of genetics. So here's Gregor Mendel. He's known as the father of genetics, and he has a pretty interesting background. So he was born in 1822, and he was living in a part of the Austrian Empire, which is now a part of the Czech Republic. He lived on a farm. He had an older sister and a younger sister, and he really enjoyed just life on the farm, but he was very interested in learning too. He had his own beehives that he kept. He did a lot of the farming and helping his father out with the farming on the land, but he also enjoyed studies. And so he went to school, and then he reached this point where his family couldn't afford to pay any more money for him to go to school. They really needed him to help out on the farm. And he didn't like that. He wanted to learn more about the world around him. He enjoyed physics, and he wanted to learn much more about the world of academia. And his youngest sister, actually gave up her dowry so that he could continue his studies. And he later repaid her by helping to support her three sons and sent two of them to medical school. Anyway, so Johann, as he was called back then, Johann Mendel, he reached this point where he wanted to keep learning more and he ran out of money. And so the only way that he could continue to be a student was if he joined a monastery and became a friar. And so he did that. And that's where he got the name Gregor. They gave him that name when he joined the monastery. And in a sense, the rest is history. When Gregor was in the monastery, he devoted his time and efforts to agriculture, specifically looking at pea plants. And he studied 7 different traits in pea plants. And, Gregor Mendel is doing all of this experimentation related to the world of genetics prior to us having a good understanding of what DNA was. They hadn't even identified nucleic acids. That term didn't exist yet in the world of science. And here's Gregor trying to figure out how traits are inherited or how traits are passed down from one generation to the next. His test subject were pea plants. And he studied, again, these 7 different traits. He looked at the flower color, the flower position, the seed color, the seed shape, the pod color, and the stem length on these pea plants. There's a couple of different stories about Gregor and his times at the monastery. And if you want to know more about those stories, just ask me about them in class. But what Gregor did figure out when he was doing his studies is that he determined that there were pairs of traits that organisms had. He called these pairs of traits factors. We call them alleles now. But he determined that each organism inherited pairs of factors from their parents. Great understanding for having very little technology. You can see in the picture here some of the different factors and traits that Gregor looked at. And you can see which is dominant and which is recessive. And again, we've already talked about what it means to be dominant, what it means to be recessive. Maybe you noticed here the seed color. The dominant seed color is yellow. And in pea plants, the recessive seed color is green. I don't know about you guys. I haven't seen very many yellow peas. Most of the peas that I eat for dinner are green. And so this is just one example that shows you that the word dominant doesn't mean common. Again, what the word dominant means is that that's the allele that when inherited, it's going to show up in the phenotype. So if a pea plant inherits a yellow allele for seed color, they're going to have yellow seeds. Even if they're heterozygous, they're still going to have yellow seeds. The other phenotypes and dominant and recessive are listed here. Later on in the problem that we're going to do, we're going to be looking at round versus wrinkled seed texture. So the first kind of genetics problem that we're going to be looking at are monohybrid problems. I know that many of you have studied genetics, but if you have or if you haven't, don't worry. I'm going to do some review of these problems and how to solve them with you now. So what makes monohybrid problems different from other kinds of problems? It's just that we're crossing a single trait at a time. If we break down the word monohybrid, You guys know what the prefix mono means. And hybrid, well, you might be thinking of a hybrid vehicle. It's a crossover. It's a combination of two different kinds of cars in terms of their fuel efficiency and what they're using for fuel. In this case, though, what we're looking at is we're crossing just a single trait at a time. Again, mono being one, hybrid being crossing a single trait at a time. All right, here's our problem that we're going to be solving today. The question is, what fraction of pea plant offspring would be wrinkled when two heterozygous round pea plants are crossed? Use a Punnett square to support your claim. So we're going to make a claim that's going to answer the question, and then our Punnett square is going to provide the evidence to back us up. The first thing that we probably have to do, though, is we're going to have to determine what's dominant and what's recessive. Now, I give you a clue in this particular problem. I tell you that we have two heterozygous round pea plants. Now, as we've talked in a previous video, the term heterozygous means that the alleles are different. But we also understand what the word dominant means. It means that if that particular allele is inherited, it's going to show up in the phenotype of the organism. So the fact that we have round pea plants who happen to have the heterozygous genotype tells us that round is dominant over wrinkled. So we're always going to have a key in our genetics problems, and our key is pretty simple. We use the first letter of the dominant phenotype to represent the alleles in our key. And so I'm going to have a capital R for the dominant allele, which we decided was round. And then I'm going to use a lowercase r, lowercase r. Oh, there we go. Okay, there we go. That lowercase r is going to represent the wrinkled phenotype for our pea plants. The parents in this particular cross, they're both heterozygous. So their genotypes are big R, little r, not F, R, being crossed by a big R, little r organism. All right. This box that we see here is a Punnett square. A Punnett square is a tool that was developed by Reginald Punnett. He was a British geneticist who lived in the 20th century. And he just developed this tool to help us to organize our factors when we're doing a cross. Now, this particular Punnett square, or any particular Punnett square, they represent some very important things. What we have going on here is we see basically meiosis and fertilization and probability all coming together in one place. We purposely studied meiosis before studying genetics because you have to have an understanding of meiosis in order to really understand what's going on here and what these letters represent and what's going on with these letters. Now, in studying meiosis, we know that organisms have pairs of chromosomes in their cells. Even a pea plant has pairs of chromosomes inside of its cells because it's the result of sexual fertilization, where two different cells came together, were fertilized, joined together, combined their DNA together to give an organism this genotype. Now, what happens during the process of meiosis is that the pairs of information separate from each other. We don't, or in the case of this pea plant, they don't pass down a big R and a little r recipe to their offspring. They either pass down a big R to their offspring or they pass down a little r to their offspring. Again, the R's are representing recipes in DNA. Instead of writing out the entire ATCG DNA code, we use just a letter of the alphabet to represent that entire maybe 300 nitrogen base pair, 3,000 nitrogen base pair long code is more easily represented with just a letter. So let's go ahead and show how meiosis works. We can take these alleles and we're going to show how the alleles are separated during the process of meiosis by drawing a single allele at the top of each of these columns. There's the big R, here's the little r from that organism. The other heterozygous round pea plant that's being crossed with this particular heterozygous round-seeded pea plant, we're going to show the separation of alleles that happens during the process of meiosis, where we have diploid cells that eventually form haploid gametes. And so we separate the alleles from each other. Again, organisms don't pass on pairs of alleles to their offspring. They pass on single versions of alleles to their offspring because of how meiosis occurs. What this big R represents up here, it represents a gamete. And that gamete has the big R recipe. It has the recipe for roundness for pea plants in it. This little r is part of a gamete. And this little r has the recipe for having wrinkled phenotype for pea plants. And the Punnett square is going to show what are the possible offspring that these two parents can produce. Now, to show fertilization, which is what we get in these boxes when we have one gamete joined with another gamete, we're basically just kind of bringing the gametes down, bringing their alleles down and over. So if fertilization were to happen between these two gametes here, the gametes may be from the father pea plant and the gametes from the mother pea plant, it's possible that they could have an offspring that is big R, big R. That's how their alleles line up and join. This big R comes down, this big R allele comes over to the right, and we end up with a genotype big R, big R. In the second box, the small allele comes down, the dominant allele comes over. We always write the dominant allele first. It's just tradition and habit and good practice. There's the potential that these parents could have an offspring that is heterozygous. Now we go to the bottom two boxes, bring down the big R, bring over the little r. We end up with a heterozygous genotype, big R, little r. And then lastly, in the last box, the little r's come over. These parents could potentially have an offspring that has the genotype little r, little r. Now, if these two parents had 4 offspring, we would predict that they would have one offspring that is homozygous dominant, big R, 2 offspring that are heterozygous, big R, little r, and 1 offspring that is homozygous recessive, little r, little r. This is our cross, this is our Punnett square. But we have to go back to the question and make sure that we've answered that. The question says what fraction, I'm going to underline that because that's key, what fraction of pea plant offspring would be wrinkled. Okay, so I'm looking for that particular phenotype when those 2 heterozygous pea plants are crossed. So what fraction of the pea plant offspring would be wrinkled? Well, you have to look at your results. Come up with your answer. It's this one here. That's our wrinkled offspring. Round is dominant over wrinkled. This offspring would have a round phenotype. This would have a round phenotype. This one would have a round phenotype. This is the only wrinkled. So the correct answer at the end is 1/4. 25% is not the correct answer because it's asking you what fraction of pea plant offspring would be wrinkled. And 25% is not a fraction, it is a percentage. All right, you've got some practice problems to do. You are welcome to watch this video at any time to give you some review to help you to understand the concepts. And if you have any questions, always please let me know. Thanks so much for watching. Take care.